There are currently well over 100,000 people awaiting organs for transplant in the United States alone. Many patients die while waiting for a candidate for transplant, while others receive the organ only to have their immune system reject it, causing the onset of graft versus host disease. Even individuals who have successful transplants typically have a limited lifespan, due to the aging and the use the organ has already endured. Imagine a world where failing organs were easily replaceable, with brand new tissues sourced from embryonic stem cells, where cancer patients requiring bone marrow transplants no longer needed a donor match to replenish the hematopoietic stem cells in their marrow. Genetic diseases, such as cystic fibrosis, that compromise quality of life in children could potentially be eliminated permanently. The therapeutic possibilities offered through stem cell research are immense. With all the potential health benefits provided by stem cells, why have we limited our research? There are religious and ethical concerns surrounding the creation and manipulation of stem cells, particularly embryonic. When fertilization occurs, the resulting zygote is totipotent, meaning it can differentiate into any human cell type. As the zygote begins to divide, it eventually reaches the 16-cell morula stage. At this point, cells differentiate into either the inner cell mass or trophoblast cells. The trophoblast layer gives rise to extraembryonic tissues, such as the placenta, while cells comprising the inner cell mass are pluripotent, differentiating into endoderm, ectoderm, and mesoderm layers. Pluripotent stem cells, commonly referred to as embryonic stem cells, can differentiate into all adult tissues, but without the trophoblast layer, they are incapable of becoming an entirely new organism. The primary source for embryonic stem cells is the zygote formed from in vitro fertilization. Many religious groups believe that life originates with fertilization, and therefore creating an embryo and inhibiting it from developing into an organism for the sake of scientific research is immoral. They see it as no different from abortion or murder, regardless of when it occurs. This is rooted in people's definitions of human beings. Do humans originate at the zygotic or blastocyst stage? Is it not until the 3 germ layers differentiate through gastrulation that life is considered human? Or perhaps the fetal stage, during brain development? Individuals' beliefs vary greatly regarding the timing of when a fetus becomes fully human, and therefore the embryonic stem cell research originating from a fertilized zygote is not universally considered ethical. Embryonic stem cells are not the only source of stem cells, however. Some, including cord blood stem cells, are extracted from discarded umbilical cords following childbirth. Rather than waste the tissue, it is kept sterile and processed in labs to isolate the undifferentiated CD34-positive stem cells. Dr. Colleen Delaney's lab at Fred Hutchinson Cancer Research Center has pioneered a method of expanding the stem cells ex vivo in culture flasks, utilizing them in transplants for immunocompromised patients. Children suffering from lymphomas or leukemias undergo aggressive chemotherapy, and rather than replenish hematopoietic stem cells with bone marrow, Dr. Delaney transplants undifferentiated stem cells for engraftment. Her research has saved many patients' lives. However, cord blood stem cells are multipotent, meaning the range of cells they can differentiate into are limited to lymphocytes and leukocytes. If embryonic stem cells could be generated in a similar way, would the research be considered moral? Or would the same individuals opposing embryonic stem cell research argue that we are flirting with the fine line between treating patients and playing God? A method has been published successfully de-differentiating human somatic cells into pluripotent cells, called induced pluripotent stem cells. Through the insertion of genes commonly expressed in stem cells using retroviral vectors, somatic cells group together and begin replicating, creating a structure similar to a blastocyst. They do not have the ability to create a brand new human being, However, they can differentiate into cells specific for many tissues throughout the human body. Does this change the ethical issues surrounding stem cell research? If stem cells can be created synthetically, without the use of fertilization, could research then be considered universally moral? Manipulating genes in regenerative stem cell research could lead to the ability to successfully generate totipotent cells. If scientists discovered a way to create human life through the de-differentiation of somatic cells, would this artificial generation be considered immoral? Even if stem cell research saved the lives of 100,000 individuals that were awaiting the organ transplants each year, where do we draw the ethical boundaries to prevent immoral consequences?
Some days her shape and the doorway will speak to me.